Welcome to the cell cycle and mitosis video for BioCore 381. The process of cell division is how a single cell fertilized egg transforms into a multicellular organism. Mitosis ensures that the genetic content of a cell, DNA in the form of chromosomes, is replicated faithfully in each daughter cell. Let's take a closer look at the cell cycle. The cell cycle is composed of four main stages, including G1 for gap1, S for synthesis, G2 for gap2, and M for mitosis. Non-dividing cells reside in G0 and can transition to or from G1. G1, S, and G2 are collectively known as interphase, which takes up the majority of the cell cycle. The stages of mitosis, which we will discuss later on in detail, are prophase, prometaphase, metaphase, anaphase, and telophase, including cytokinesis. How long does the cell cycle take? Across different species and different cell types, even within the same organism, there is a large range of cell cycle durations. For example, budding yeast can complete a full cell cycle in only 90 minutes. But let's consider a proliferating adult somatic cell with a cell cycle of 24 hours, such as a fibroblast. In this case, G1 might last 11 hours, S 8 hours, G2 4 hours, and M only a single hour. Next, we will examine each stage in detail. In the nucleus, each chromosome is loosely organized during the stages of interphase, that's G1, S, and G2. The chromosomes probably look something like this and would be impossible to tell apart, even under a microscope. But for ease of drawing, I'll be depicting the non-condensed chromosomes with outlines. Here, I'm showing 4 chromosomes, a long pair and a short pair, each with one maternal chromosome in red and one paternal chromosome in blue. In humans, the cell cycle begins with a diploid cell that contains 46 chromosomes, 23 maternal and 23 paternal. That's 22 pairs of homologous chromosomes and 1 pair of sex chromosomes. The genetic content of this cell is 2N because there are 2 homologous chromosomes, one paternal and one maternal. The chromosome content of the cell is 2C, where C depicts a full set of 23 chromosomes, one from each parent. Now, let's go over some of the important structures and nomenclature. Chromosome, or dyed body, is so named because of the cell dyes that early scientists used to visualize chromosomes during mitosis. Although the DNA sequence of every chromosome is different, they all contain specialized DNA sequences, including the centromere, where the kinetochore proteins bind, and telomeres, which are located at the end of the chromosome. Outside the nucleus, the cell has a microtubule organizing center, or MTOC, or MTOC, which is called the centrosome in animal cells and is made up of a pair of centrioles. If all these terms are hard to keep straight, keep in mind that some means body, aka a visible structure, and mere means part. Now let's return to the cell cycle. In gap1, the cell prepares for cell division. The cell continues to grow and must pass through a number of checkpoints before it is committed to division. These checkpoints include nutrient availability, signals from other cells, and cell size, among other factors. Let's move on to S, or synthesis phase. In this phase, the DNA of each chromosome is copied and packaged with its accompanying structural proteins, such as histones and the kinetochore. At this point, the genetic content of the cell is still 2N because no new information has been added or taken away. But the chromosomal content is now 4C because each chromosome was duplicated. Each chromosome is now a pair of sister chromatids, though it is still referred to as a chromosome, a dyad, because the chromatids are still connected to each other, still a single unit. Therefore, even after DNA synthesis, there are still 46 chromosomes, though there are 92 chromatids. In G2, the cell synthesizes the proteins needed for mitosis and passes through additional checkpoints, such as confirming that all chromosomes have been duplicated. The genetic content and the chromosome content remain unchanged. Finally, we get to the first stage of mitosis, prophase. In prophase, the loosely coiled DNA begins to condense into observable chromosomes like those seen in a karyotype. I will depict each chromatid as a thick line. In addition, the nuclear membrane begins to break apart, and the mitotic spindle begins to form from the Mitosis, or the centrosomes. In prometaphase, the nuclear membrane completely fragments, and the centrosomes move to opposite sides of the cell. The mitotic spindle is now fully formed. The spindle poles, which include the centrosomes/MTOCs, anchor three types of microtubule connections. Kinetochore microtubules, in green, connect the microtubule organizing center to each chromatid at the kinetochore and centromere. Polar microtubules connect each spindle pole, and aster microtubules anchor the mitotic apparatus to the rest of the cell. In metaphase, the kinetochore microtubules from the spindle poles begin a tug-of-war with each pair of sister chromatids. The spindle poles are equally matched and eventually pull each pair of sister chromatids into alignment on the metaphase plate. The genetic content and the chromosome content remain the same. In anaphase, the connection between the sister chromatids is degraded and the chromosomes are pulled apart towards opposite sides of the cell through the shortening of the kinetochore microtubules and the lengthening of the polar microtubules, which push against each other. The chromosome dyad becomes two chromosome monads. For this brief window in time, there are 92 chromosomes in the cell. During telophase, the nuclear membrane reforms to generate two separate nuclei, and cytokinesis separates the cytoplasm into two individual daughter cells. Each daughter cell now has a 2N genetic content identical to the parent cell. Note that the genetic content remains constant throughout the cell cycle in mitosis. And each daughter cell now has a chromosome content of 2C, or 46 chromosomes in humans. What I've shown you here is only the tip of the iceberg as concerns the cell cycle in mitosis. And a lot is still unknown about this process that is so vital to multicellular life. If you're curious, there's much more to explore about the topics listed here.